Welcome to Research Conference. Let's go to the first slide. Yeah, so I have your title. I'd like to welcome Dawn Bowles, Assistant Professor in Surgery in the Surgical Sciences Division, and she is also co-director of the Duke Human Heart Repository, which is the topic of the talk today. She has extensive background in microbiology, particularly in virology and viral vector development, which is out of scope for me. Dr. Bowles has a BS in microbiology from the University of Louisiana, Lafayette, PhD from Louisiana State University, and working with herpes gene regulation. Postdoc, she was over at UNC with Jude Samolsky in the Gene Therapy Center. And currently, she's at Duke, research focused on discovery and testing and translation of novel therapeutic modalities for cardiac injury and end-stage heart failure. She works with Carmelo Milano, who's also on the faculty in the DCRI, to develop a human heart repository that serves as a discovery engine for these laboratories. So I think this is what we're going to hear about on this laboratory resource. Remember, questions, wait for the mic, and because we do have some people at Durham Center, and we are streaming this for archival purposes. Thanks. Dawn?
For the introduction. Can everyone hear me? In terms of questions, I'm perfectly comfortable taking questions throughout the talk. In fact, I'd prefer that if possible, so feel free. So what I want to talk about today is a resource that we've been working on for quite some time in the laboratory, and we've been, in the past 4 or 5 years, developing it as a Duke resource. It's the Duke Human Heart Repository, or DHHR for short. And what I'd like to do in this talk today is to go over 4 different topics. One is an overview of different academic and commercial cardiovascular tissue banks in the United States, and to prove to you why we're necessary. Then I want to describe a little bit about the current cardiovascular tissue banking efforts that we're doing at Duke, letting you know what we have available and how to contact us and what we can potentially do for you and with you. I want to describe some of the studies that we're doing in the laboratory that the DHHR is supporting, and maybe give you some ideas about what we can do with you for projects. And then I want to discuss some of the growth and sustainability plans that we have planned and in progress, and perhaps get some feedback from you in terms of whether this is an appropriate path or whether there are some other things that we should be thinking about. So Gail Henderson at UNC quite recently published a survey, a national survey of biobanks in the United States. And she was addressing a problem that I've run into, in that there seems to be no consensus or census or registry of biobanks in the United States. So their group compiled a list of 636 US biobanks, and part of their exclusion was to eliminate banks that had no aim to share their biospecimens. And a requirement for inclusion in the study was a web presence, so there had to be some sort of easy way of getting information on these biobanks. And they were able to recruit 456 banks for a 30-minute online survey. And some of the information that was acquired from this survey was that about 75% of the banks that are in the United States have been established in the past 10 years. Most people worry about underutilization of their biobanks, and one thing we're really worried about is long-term funding for sustainability. The majority of the banks have focused on cancer, followed by neurological banks such as Alzheimer's, and then HIV/AIDS. Most biobanks have a small number of specimens from each tissue source. Serum and plasma are the most common specimens, followed by solid tissues. And not mentioned in this paper, but a personal communication from Dr. Henderson, was not many banks were banking human heart tissue. Suresh Balu, sitting in the back of the room, did a systematic study of the different biobanks in the United States, and he was able to determine that there were 7 major heart repositories in existence in the United States. And these are our peers: Penn, Cleveland Clinic, Vanderbilt, and 3 more on the next slide. And what I'd like to point out is the premier example would be at Penn. It's led by Dr. Ken Margulies. And in their bank, on their website, they advertise that they have 850 human hearts that they've banked and about 18,000 specimens. And this bank has provided tissues to 35 academic laboratories, and resulting from this has been at least 46 peer-reviewed publications. The other banks are not as established as the one at Penn. Brigham, Stanford, UVA has an aortic aneurysm specimen bank, and then also UCSF. Many of these tissues, our heart tissues, are acquired from surgical procedures, such as transplants, and also from discarded pieces of tissue from different surgical procedures. I want to mention at Brigham, on their website, as of 2012, they mentioned that they aim to collect 200 hearts. Suresh also looked at the major commercial biobanks to see who had heart tissue. And a few of them did, but there was a high price that was required to be paid in many cases in order to obtain these kinds of tissues for research. So together, this information establishes that there is still a need for banking human heart tissue. There's not much available in the United States for research and discovery. And so with this information, we're going forward with the Duke Human Heart Repository. And this is an overview of what we are and what we do. We acquire hearts and heart tissue from 3 different sources: from explanted hearts from transplant procedures, from donor hearts from different organ procurement organizations. These would be our non-failing control tissue that we acquire, and then from other surgical procedures. Associated with the hearts and the heart tissue are clinical information that is searchable. In the laboratory, we do multiple different things with the heart. We either process them into different kinds of cells or tissue sections that we use for research, or we also do experiments using viable heart tissue. And I'm going to show an example of studies that we're doing in the laboratory using whole hearts, acquiring functional data and functional information from these hearts. But we're more than just a bank of tissue. We're more than just tissue sitting in the freezer. We are capable and actively perform all these different kinds of procedures. So Duke is a high volume center in terms of both transplant and open heart procedures. And this is a summary of the number of heart transplants that have been performed at Duke over the past 5 or 6 years, and also the VAD procedures that are performed at Duke. And all of these procedures would yield tissue for research. We are number 4 in the United States for transplants in terms of volume, and we are pretty high also in the country in terms of the number of LVADs that are performed. And over 908 transplants have been performed at Duke since 1985 when the program first started. So from these kinds of procedures, there's the potential for thousands of samples on a yearly basis. We also have relationships with organ procurement organizations. These are numbers that were obtained from the Regional Organ Procurement Organization, Carolina Donor Services, from the years 2009 and 2010. And what's important to note is of all the hearts each year that are evaluated for heart transplantation, really only about a quarter to a third are transplanted. Many of the remaining hearts are available for research purposes. And in 2009 and 2010, we acquired over 80 of these human hearts for our research endeavors in the laboratory. So currently in our human heart repository, we are staffed by the following people. Dr. Milano and I co-direct the facility. We have a repository manager, Mike Watson, who's sitting in the audience, a lab analyst, Raj, and we also have surgical residents and postdoctoral fellows who are all part of what we're doing in terms of the services that we offer. And as of yesterday, we have in our repository hearts, over 370 human hearts have been collected over the past 4 years. We have additional historical samples that would make us more competitive in terms of the numbers that Kim Margulies has, but these were done, these were obtained under very rigorous, rigorously detailed protocols. And we've estimated that we have about 36,000 specimens. From every heart, we acquire multiple specimens from many different areas of the heart. And these samples are flash frozen, they're formalin fixed, they're put into RNA later, OCT embedded. Associated with this are thorough clinical information, including, in many cases, echoes. And we have obtained from the different OPOs specimens from approximately 100 non-failing hearts, which would be controls. We have a relatively high sample diversity, ischemic and non-ischemic cardiomyopathies, the non-ischemics from many different etiologies. Pretty much equal representation by gender, a wide age range from 6 months to 88 years. And we're supporting the research projects of 16 Duke investigators currently and 4 external collaborators. And this is part of our ongoing awareness campaign, trying to make more people here at Duke aware of this resource and what it can do. So the next set of slides is going to be examples of what we have done in the laboratory with these heart samples. And because of our very large group of samples that we have, we were able to obtain, I'll be able to see this a little bit better. So what this set of experiments is showing is a mass spec proteomics analysis of human heart samples from 3 different groups with a sample size of 4. The groups were non-failing human hearts, ischemic failing, or non-ischemic failing. And because we have a very large repository, we were very able to finally match these samples in terms of age. They were all in their mid to late 50s, sex, all male, matched according to race, and whether they had prior bypass surgery in the case of the ischemic failing, their cardiac function by ejection fraction, the types of inotropes, or whether they were on inotropes, and whether they had some other kind of intervention. We were able to acquire or utilize these samples, and in conjunction with the Duke Proteomics Core, they were able to perform an unbiased open platform proteomics assessment or an enrichment for the phosphoproteome. And this is just showing the workflow that was done where the samples were extracted in trizol and then 25 micrograms was spiked with yeast and then subjected to label-free mass spec, or a portion of the sample was subjected to phosphoproteome enrichment with titanium oxide columns, and then subjected to label-free mass spec. B is demonstrating the reproducibility of the mass spec by looking at the spike. And then C is a summary of what we found in terms of the numbers of proteins. And from all 12 samples, we were able to both qualify or identify and quantify 850 total proteins that we could compare the relative levels of them to each other in these different samples. A summary of this data, or a summary of the proteins that were statistically significantly different with a greater than twofold change in the ischemic failing versus non-failing shown in the blue, or the non-ischemic failing versus the non-failing controller shown in the red. And what you can see is that for the most part, there weren't a tremendous number of proteins that were differentially up or down regulated, which is it shows the quality of the tissues that we have, the high quality nature of the tissues, but it also gives us some nice molecules to look at further in the laboratory. The next slide is showing a summary of the fold changes of the statistically significant proteins that or peptides, I should say, that were identified in the phosphoenrichment of the ischemic failing versus non-failing in blue, and the non-ischemic versus the non-failing in red. And again, not a tremendous number of proteins were differentially phosphorylated, but interestingly, several of them, for example, pyruvate dehydrogenase was very differentially phosphorylated, highly significant at 3 different sites in the protein. So molecularly, these are some candidate proteins that we want to follow further in our studies and our analyses. So because from each heart we acquire so many pieces of tissue, which really isn't seen in cancer tissue biobanking, because cancer tissue is limited by relatively small sizes of tissue. Kind of when you do these kinds of studies, you don't have much sample to go back to look at. But in our case, we were able to actually go back to replica samples of the same hearts and do some validation studies. And these are showing examples of Western blot studies that we did, looking at ischemic failing hearts, the non-ischemic and the non-failing, and using antibodies to different proteins that we identified in our proteomics analysis. We're able to validate our proteomics findings. And some of these things are quite interesting proteins that we are planning to examine further. For example, these fibrillin 1 and fibrillin 2 proteins are involved in extracellular matrix remodeling, and they were upregulated in both the ischemic and the non-ischemic failing. We're also interested in this alpha-2 macroglobulin, which appears that cleavage fragments are increased in the ischemic failing and the non-ischemic failing hearts. And these are things that we're trying to develop further and following through in many of our other 374 hearts to see how universal these findings are. But from this type of mass spec proteomics analysis, we can generate a large data set. And these are examples of some of the bioinformatics inquiries that we've done, putting these different players that we found that were differentially changed into an Ingenuity pathway analysis software program. And some of the things that came out are not surprising. Pathways that seem to be perturbed in these hearts were fibrosis, pathways involved with Marfan syndrome, abnormal collagen organization, tumorigenesis, and the oxidative stress response. But importantly, some of the things that it enables us to do are to dig further deep into the pathways and come up with additional candidates that we can explore further in the laboratory. So what I showed in the previous slides were examples of an unbiased assessment of our samples where we really had no idea of what we were going to find. And this is very nice for discovery purposes, but there may be some instances where you might have a hypothesis where there's a protein that you might be interested in that no one knows about in the literature, that no one's ever looked at in the context of heart failure, heart disease. And this repository would be an ideal place for you to go to gather some preliminary data. And this paper that came out of our laboratory is an example of one of these things, how we interrogated the repository to look at expression of a particular protein involved in apoptosis, or the XIAP protein. So what we knew was that the XIAP protein was a potent inhibitor of apoptosis, and that there's a vast literature showing the importance of the involvement of apoptosis in heart failure and heart disease. And so one of the things that we were interested in was, what about these inhibitors of apoptosis? What happens to their levels, their fate in human cardiac tissue and human disease? And XIAP is the most potent inhibitor that's been described in the literature. And it binds to the catalytic sites of these terminal caspases in the caspase 3, 7, and 9. So it's a very potent inhibitor. And so looking at the literature, this is an analysis of 11 publications that looked at different apoptotic proteins in human heart failure. And looking at what was reported from 1996 until 2009, there seemed to be some controversy in the literature regarding XIAP levels. One paper noted that XIAP levels diminished, and another paper noted that XIAP levels increased. And so we were interested in what's the case? What do we find in our hands with our repository? So we did a series of studies where we specifically looked for XIAP using either Western blot analysis with XIAP antibodies or a focused apoptotic array looking at the levels of different apoptotic proteins concurrently. And what we found was that in our non-ischemic failing hearts, we found a diminishment of XIAP protein in both the Western blot analysis in the failing hearts and using our apoptotic arrays. We found a diminishment of XIAP. And then concurrently, with the same samples, lysates from the same samples, we're actually able to do a very sensitive caspase 3 activity assay based on luciferase from Promega. And we were able to show that concurrent with the diminishment of XIAP in the failing hearts, there was an increase in caspase 3 activity. So this information fed into my interest in the laboratory for gene therapy viral vector development. So I went ahead and devised a viral vector, adeno-associated viral vector, that overexpressed XIAP in a cardiac-specific manner. And then did a series of studies, and these studies were done by Valentino Piacentino, one of the surgical residents when he was in the laboratory. And this is using rat neonatal cardiomyocytes, sort of a workhorse of cardiac cells in the research laboratories. And the cells were infected or transduced with either a viral vector that expresses GFP or green fluorescent protein, or the viral vector that expresses this XIAP protein. And what we found was, upon induction of apoptosis by chilithirin, isoproterenol, or hypoxia ischemia, we were able to demonstrate that overexpression of XIAP is cardioprotective. So these studies are enabling us to go forward to further develop XIAP as a potential therapeutic, and things that we're planning to do with XIAP are to look at the universality of this observation in all of our human heart samples. We're trying to develop a high throughput assay where we can specifically look at the relative levels of XIAP across everything that we have to see, is it just in the non-ischemics, or are we seeing it in the ischemics also? We're planning to develop a transgenic mouse model that will overexpress XIAP specifically in the heart and determine whether it can be cardioprotective, and then also do a series of studies using AAV-XIAP gene therapy. But our repository provided us with some very nice preliminary data to go forward. And so these are, this is a route in which investigators can use this repository if they have interest in specific molecules, protein, RNA, metabolite, that sort of thing, in a targeted way. So, switch gears a little bit, and I'll describe a series of studies that we've done in the laboratory based on our abilities to acquire intact human hearts and to acquire functional data on human hearts ex vivo. So this is a slide that I got from Dr. Milano showing that an important clinical problem is that of all the hearts that are evaluated for transplantation, only about a quarter of these hearts are transplanted, and the remaining hearts are not transplanted. The most common reason is a concern over organ quality. You just wouldn't want to transplant something that you're unsure whether it will function at the end of the procedure. So there are two main donor types for transplantation in general. There are donation after brain death and donation after cardiac death. And the only donation after brain death donors are utilized for heart transplantation. Donation after cardiac death, or DCD for short, these are being utilized for other organs such as the liver, the lung, the kidney, but because of ischemic damage that happens following cardiac death, there's a concern that these hearts may be irreversibly damaged. And one of the projects that we have ongoing in the laboratory is to expand the donor pool by specifically evaluating function of DCD hearts that we acquire. So to say that DCD donors are potential, the first human heart transplant that was ever performed was actually performed using a DCD donor. However, it's very rare that a donor would be at the same location as the recipient, and a lot of the success of cardiac transplantation involves a transport of the organ. So we don't normally there's about a 2-hour transport period, which could lead to more damage happening, I guess. But this is our experiences with the human DCD heart. And through our relationships with the different organ procurement organizations, we're offered these hearts for research. And this is a human heart that underwent a warm ischemic time of 13 minutes, and then it was transported back to the laboratory in about 3 hours. So we have a total ischemic time of over 3 hours. And in the laboratory, we have a Langendorf working heart apparatus. This basically is a situation that mimics a transplant, where we can put the heart into a circuit where it's perfused with blood, and it's eventually shocked, and it starts to work if we're lucky. And this video that I'm going to show you is this heart, actually a DCD heart, human DCD heart, that's functioning in the laboratory 3 hours after it's been removed from the donor. And what I would point out right here is that this has not been reported in the literature before, that a heart that long, a DCD heart that long after removal actually performing some work. Now, whether it's performing work enough to support life, that's the question that we're trying to find out. And we're trying to develop a protocol. Well, this is an example of the pressure volume loops that we can obtain ex vivo. So this gives us an indication of the degree of function that we have on these hearts. But what we're trying to do in the laboratory is to develop a protocol that will enable us to predict the function of a DCD heart post-reperfusion on an ex vivo circuit. And what our protocol relies on is, one, our access to these types of hearts, but two, instead of doing cold static storage, which is standard of care, we're using a perfusion apparatus, a cold perfusion apparatus, which the hearts upon procurement are connected to this apparatus, and then during this preservation period, we're acquiring from a port here the perfusate that is being that's going through the heart, and then we're assessing different biomarker levels like troponin I, troponin T, lactate, hypoxanthine, CPK and B, to understand the degree of damage that's happening or has happened to the heart. And then after this 2-hour storage period, we're putting the heart onto the apparatus in a sort of a resting mode, increasing the temperature, mimicking what happens during a cardiac transplantation. And then we're putting the heart into a working mode and assessing ex vivo function with PD loops. And what we're hoping is that this information that we're acquiring during the storage process will enable us to predict how this heart functions post-reperfusion, so that we can predict clinically, in a clinically relevant time period, which hearts are going to be, or DCD hearts would be usable for transplantation and which ones would not be usable for transplantation. But this is just an example of some of the things that we can do, or one of the things that we can do with our experiences with viable human heart tissue in the laboratory with the repository. So the fourth project that I want to describe is something that has been a recently funded project. We find it quite exciting in the laboratory. It's funding from NASA, and we could not have gotten this grant without access to this human heart tissue. One of the problems that NASA is concerned about, especially in their plan to go to Mars in the near future, is that if the heart stops working, if the astronaut's heart stop working during the trip to Mars, it's going to be at least 18 months there and 18 months back. This could result in the loss of a mission. And they are unclear as to what the long-term influence of different things such as microgravity and radiation, how that will impact the astronauts to and from Mars. And so what we're doing with NASA is that our working hypothesis is that changes in the protein profile of cardiac tissues following exposure to microgravity may provide information on mechanism of damage, predictors of damage, and potential countermeasures that could happen or that could be employed. And so we were provided this bioreactor from NASA, and basically what it does is it mimics the effects of microgravity on cells. And we are undertaking a series of experiments, first by putting rat neonatal cardiac cells in this microgravity reactor. And it basically mimics the vomit comet that astronauts go up in training during part of their training process. And it's a free fall, constant free fall. These cells are in constant free fall in this rotating device right here shown in an incubator. So we're doing these experiments with both rat neonatal cells to work out all of our protocols, and then we're going to move on to human cardiac cells and tissues to see what the proteomic and phosphoproteomic changes, which if any changes are happening as a result of microgravity. This series of experiments is in progress right now, so I don't have any data to report on right now, but we find it to be a very exciting use of our repository in the laboratory right now. So I just want to switch gears and talk about some of the sustainability efforts that we're trying to do. We're anticipating to submit several grants in the near future. One I haven't mentioned any data about, but we're working with Dr. Nair, who has done quite a bit of work with RNA dendritic cell vaccines. And one of the things we're interested in is reprogramming fibroblasts into cardiac progenitor cells using RNA. And because of our repository and our access to human cells and tissue, we're able to isolate human cardiac progenitor cells in the laboratory and evaluate them and use them as controls, as well as using cardiac cardiomyocytes in these types of studies. And we're planning to put together a grant application in the near future. I mentioned the XIP project. We are moving that forward further, but the repository will be heavily highlighted in this grant application. Some of the other grants that are in discussion are an NHLBI systems biology grant. Because we acquire so many pieces of tissue from each and every heart that we have, we're really not at a loss for substrate. And so what we feel we can do is we can evaluate numerous types of omics: transcriptomics, genomics, proteomics, metabolomics, epigenomics, and we can develop a framework, a very systematic overview of what these different types of disease states look like at the systems level. And this is something that we're actively working forward. There's also an RFA for transplant and following transplant outcomes, and we feel that our repository will play a role in this. But the major grant that we're going for is an R24 infrastructure grant, and I just want to discuss this in a little bit more detail. So NHLBI has made an announcement for resource-related research projects in R24. And one of the items that they're interested in for the NHLBI community are novel biorepositories for heart, lung, and blood disease. And based on what I've told you, we feel that we are or can be a novel biorepository for the heart. And what we want to do is to write a proposal that synergizes the expertise of surgery, the DTRI, and the strong omics course here at Duke, and transform our institutional human heart repository into a national resource that is rich with clinical and molecular information and is accessible to a wide array of cardiovascular investigators, not just here at Duke, but nationwide, and something that is financially sustainable. And that's a key ingredient in this proposal. NHLBI absolutely requires that you have a financial sustainability plan. But we think that this resource will allow many more investigators unparalleled opportunity to answer some key questions in human cardiac physiology. open up some of the things that we're beginning to touch on with our DCD project and with our NASA project. So I have 3 aims, and very briefly, we want to expand and add value by doing 4 of these things. We want to better define our workflows and adopt better standard operating procedures. We want to work with the Duke Biobank and use, and we have plans in the works to use this new enterprise-wide biospecimen information management software, LabVantage, that's going to come on board for all the biobanking efforts here at Duke. We want to establish a systems biology-based information framework by leveraging these strong omics cores to provide molecular data on these human heart tissues. And then we want to further develop some additional molecular cellular tissue derivatives, such as having on hand RNA, DNA, protein, different cells that we can provide to investigators and other kinds of derivatives, as well as develop these cell and tissue-based assays on a per-needed basis for different projects. We want to disseminate the tissues, derivatives, and the information to U.S. investigators by first developing an awareness campaign. This is my first effort really at an awareness campaign of getting this information available to the consumers. We also want to create a peer-reviewed project-based system so that if someone has a good idea and might want to use our repository but doesn't have any funding yet, we'd like to be able to provide some vouchers for them to acquire these samples free of charge, but also to provide some pilot project funding through this grant so that they could enable them to gather needed preliminary data for further grant applications. And then our aims to achieve financial sustainability are to establish a viable cost recovery model. This is mainly aimed at the academic investigators, seeking more government grants than what we currently are, but looking also for industry-sponsored research agreements where industry may be interested in having access to our cells, our tissues to test different types of pharmaceuticals or ask other kinds of questions, and also philanthropy. We think that perhaps developing some of the intellectual property that we have been finding from our discoveries from our proteomics analysis might be a way in which we can also augment our financial sustainability. And then through the vouchers and the pilot funding, we think that we might be able to invest strategically into research portfolios of investigators who would then want to use the National Repository for further funding, for other mechanisms of funding. So, just in closing, if you have any questions or if you feel like these are things that these tissues or cells or access to these things might be something you're interested in, please contact me. Any good ideas for grant collaborations? This is my contact information. I also want to note that we are in the process of being indexed in the Duke Index of Biospecimens, which is a Duke-wide effort to have an understanding of all the different biospecimens at Duke. So this will be a searchable entity soon, if not already. And then I want to acknowledge the different people who have contributed to this project. Dr. Milano, who has provided the majority of the human hearts and the tissues, our repository manager, Mike Watson, and Raj, many of the surgeons and surgical residents who have worked to procure these different human hearts over the years, many of the people working in the lab doing different experiments and different projects. I want to thank the Duke Proteomics Core, who's worked with us to help evaluate these human hearts. When we first approached them, Arthur Mosley had not worked with human heart tissue before, so it was his first foray into that. And since then, we've done additional projects with him, and we've increase the number of proteins that we're able to examine. I'm not presenting that data, but we have refining methods with them. Thank the DTMI. Grindel was here. She was helping with the business development. Suresh in the back, who's helped considerably with this. Virginia Burns when she was here. And then also funding. Some of this was funded through DTRI pilot funding, especially for the DCD project. The Children's Cardiomyopathy Foundation, Duke Surgery supporting the efforts of Mike and Raj for expanding our repository, and then NIH and NASA funding for some of these other projects. And that's it.
Thank you. It's very impressive project. Thank you so much.
Thank you.
I have a question. I just wondering, you are collecting tissue.
Yes.
In the meantime, are you collecting also plasma? And my next question is, if you are collecting plasma from the same donor, do you have any collaboration with the group who works on the plasma?
So that is an excellent question, and I was hoping that that question would come up while I was here. So we are currently not collecting plasma, but that is definitely something that we want to aspire to in the future. We want to be able to acquire serial plasma samples, or as many plasma samples as possible, because a lot of our work in our tissues are discovery, but the plasma is really where we really need to go, especially to hone in, the biomarker and the biomarker development. So that would add so much value to our repository. And if you are a person who could provide guidance on that, or if you could point me in the direction of someone who could provide us guidance on how to do that appropriately, best practices, then I would definitely appreciate having a conversation afterwards with this.
Thank you so much. I used to work some group, I would like to contact you. Thank you so much.
Thank you.
Thank you.
Kevin Mahaffey, Cardiology here. A nice presentation. It's great to see this up on the screen to think through many of the issues. It wasn't until you got to the end where you're looking at the aims for your grants that I really started to get an idea of what you wanted this group to become. So maybe you could rewind for us, and rather than, I'm thinking more as a businessman now.
Okay.
Because that's what it seems critical to you. You've got to come up with a viable business strategy. And simply collecting and sharing these samples, and in fact, I thought I heard you say you were going to share them for free to some people.
Well, I mean,
but let me finish. And so, start high picture. What is your vision of this group? And then we can think through how you're going to get there. And I'm not sure I understand the vision yet.
Maybe I'm not sure I understand the vision either at this point. Our main goal is to be self-sufficient. It's to not necessarily make a profit, but to continue on with what we're doing and not go into the red. But a lot of these individuals, or all of these individuals who have provided these samples, the intention is that these are to go for research. So we don't want them to languish in the freezer. We don't want them to not do what they're intended to do in the time in which they're intended to do so. I do want to get these in the hands of the best people who can do the best work with them to make the best discoveries possible.
So and maybe Suresh can comment, because he's probably thought a lot about this, because it sounds like you have two major, marketing strategies. One is you are a group that can do the research on these. Well, you can collect the specimens, so simply collecting and storing. Mm-hmm. And then secondly, you can also do research on the specimens.
Right.
And how you approach those two from a business model perspective to fund it acutely and make it sustainable long term could be very different. Mm-hmm. I don't know, Suresh, if you've thought about those things.
Yes, when we started off, we primarily wanted to grow our assets being primarily faculty and research staff. So from that regard, we wanted to use these assets, which was already developed and maintained, going to pharma industry sponsors to primarily look for early phase research, biomarkers and all earlier compounds. Can you use the tissue and tissue samples to really make an impact in terms of speeding up the research aspects of it? That was the primary aspect, bringing in industry research, combining that with the government-sponsored research as well to really make it self-sustaining. That's the whole plan.
That's terrific. And I'm just trying to think of how DCRI could perhaps help you. And one thing off the top of my head, and maybe others in the room could share some ideas with research they're doing, is a lot of the registries and research collaborative groups that we work with see patients that unfortunately die. Like the Society of Thoracic Surgeons, the STS database is here. And whether or not if your goal is really to amass the largest repository of hearts, then developing collaborations with them where often their patients die, unfortunately. Well, what a great opportunity for you to collect additional heart samples that aren't just necessarily across the street on the third floor in the hospital. We also work here with the race investigators across all of North Carolina, the acute MI registry and initiatives. Unfortunately, many of those people die as well. And so linking in with those investigators may increase opportunities for harvesting hearts to contribute to the repository. Those are just two ideas that I have, and I'm sure others in the room will be able to help you as well. But this is terrific. Good luck.
Thank you. Thank you.